All right, so the key part of what we're going to have a look at now is the actual mitosis that occurs. If you think about what we've just talked about with interphase, that's actually primarily a large chunk of the amount of time in cell division. Mitosis takes next to nothing comparatively, and mitosis is literally just the splitting of what was just replicated. So in a 24, if we thought about this as a 24-hour process, the mitosis, the prophase, metaphase, anaphase, and telophase would actually take about one of those 24 hours, where the interphase, the replication, all the checks that occur, all of the pre-protein creations, the nutrient creations, the organelle creations, and stuff like that, and the replication would be the other 23 hours before we actually go about dividing the cell. This process does not take 24 hours, but it's just relative time, just for you to think about. So in interphase, we did talk about the development of organelles, and one of the key organelles that is created or replicated is the centrosome. And so the centrosome is replicated because we need two of them so that the centrosome can do its job. And its job is to be able to create these things called the spindle fibers or the mitotic fibers. So sort of like rail networks at either end of what will become the two new cells. So these rail networks, these fibers, can attach the chromosomes that have been duplicated and split them and pull them apart, and they can be pulled along the spindle fibers that were created. So prophase is all about the creation of this mitotic spindle, or these fibers, or think about them as rail networks. Don't talk about them as rail networks in an exam. Use the term mitotic spindle if you're talking about in an exam. And the fact is that the role of these is to be able to provide alignment and a space for the chromosomes to be able to be securely pulled apart. So a really good graphic to go along with your notes here is that one in the bottom right hand corner there. So that's sort of what we're trying to achieve here, and you can see in the middle you've got the sister chromatids, or the replicated chromosomes, and then they're going to be separated and pulled apart and pulled along those mitotic spindles, or the microtubules as we can otherwise refer to them as. Alright, so when we get to metaphase, this is now where we start to see a bit of an organisational game. So what happens here is we've got the sister chromatids will start to join to the microtubules, or to the spindle fibres. And when they join there, that's really, really important that they join at the centromere, which is the point or the middle point of the sister chromatids, or the chromosome that has been replicated. And so it basically that pierces straight through the center, and then eventually they can be split apart. And so if we have a look here, we say that they align on the metaphase plate. Just think about that as the central point between the cell that will become this sort of pinching point of the two new cells. And we can sort of see, if we zoom in a little bit further, you can see the spindle fiber, or the microtubule, that feeds right through there, and it feeds right through the actual duplicated chromosome straight through the centromere. So that's the real key thing here, is the alignment along the metaphase plate and the attachment of the chromosomes through the microtubules, or through the spindle fibers, through the centromere that exists on the sister chromatids. A lot of big words, so make sure you keep rewatching this if you need to. In anaphase, that's where we now get to be able to see the separation of the sister chromatids. And so you can see that those two that were once joined like this after they were replicated have now been pulled apart, and they're pulled along the actual spindle fibers. So now we can actually refer to these sister chromatids now as daughter chromosomes, because they represent the individual chromosomes that will exist in the new cells. And so you can see anaphase is really key. You've got that separation of the sister chromatids now into what we call daughter chromosomes. And in telophase, that's the last point where we're starting to develop the two respective nuclei, or the two nucleuses for the two particular cells. We then start to get a cleavage furrow, or a little pinch point, where in cytokinesis the cells can split apart. So telophase and cytokinesis is the development of nuclei and the splitting apart. So really, really good picture to have in your notes. and then just a couple of other slides about the overview of what's going on here as well. That'll do.